La diffusion vient de démarrer. Tous les participants sont en mode écoute seul.
All right. Good morning, everybody. So I'm Matthieu Pesant, product manager and scientific support specialist for the NGS, and that's a real pleasure for me this morning to give you a presentation on our solutions for challenging samples. So the agenda of the presentation will follow this outline. Basically, we will have two parts. The first one dedicated to solutions for RNA-seq, starting with single-cell mRNA-seq, then solutions for degraded low-input RNA. Then we will have an overview of more specific solutions that we have for small RNAs, targeted RNA sequencing, as well as immune profiling, such as TCR and BCR. And then the second part of the presentation will focus on DNA-seq application, mainly focusing on low-input degraded DNA samples, as well as single-cell solutions that we have for DNA-seq. So let's start with this global view of what we offer, and with the first part of the talk, RNA-seq. So our RNA-seq solutions are all centered around the smart technology that we developed years ago, and that basically is used for any kind of samples that you may face. So from single cell to very low input RNA quantities, degraded RNA, such as coming from FFPE samples, cell-free RNAs, virus and bacteria, but also some more specific and also challenging in terms of quantity and potentially also in terms of quality. We also have implemented the smart technology for studying biomarkers like small RNAs and microRNAs, and TCR and BCR profiling. So all our products centered around the smart technology, thanks to this technology, show really a very high sensitivity and reproducibility, and this is why these products are really well fitted for analyzing inputs that are very low, so from very limited quantities, but as well as degraded samples. The strength of the smart technology is that it is it allows the full length and complete representation of the cDNA pool that you will produce from your sample, and it is also very fast and an easy workflow because it is a single tube protocol, so avoiding loss of the material, contamination, and further degradation. So basically, the smart technology now for years has been the gold standard for what we call ultra low input, but also for single cell RNA-seq analysis. So we spoke about smart technology. So this is the main workflow that is used in our RNA kits. So SMART technology, what does it stand for? It stands for switching mechanism at the 5' end of the RNA template. So how does it work? So starting from RNA, we will first produce the first strand cDNA by priming the reverse transcription with SMART6 CDS primer 2A. So this primer is blocked to prevent further adapted dimer formation. So the reverse transcriptase has a very particular feature in that sense that it will retrotranscribe the RNA, and when it will reach the 5' end of the RNA, you will have this so-called template switching mechanism of the reverse transcriptase. It will first add those X nucleotides, which are extra nucleotides at the 5' end of the RNA, and then using what we call the SMART oligonucleotide present in the reaction, you will see that those X nucleotides from the SMART oligo have complementarity to these extra nucleotides added, so that it will bind this sequence, and then with specific blocked primers, it will allow to further synthesize the double-strand cDNA complementary to the total RNA. So the point here is that full-length double-strand DNA will be produced with blocked extremities, so just to avoid adapter primers dimers. So this main technology is used in our single cell kits. So I call them single cells, but they are also called ultra-low input RNA-seq kit. So the last version of the SMART-seq V4 ultra-low input RNA-seq kit so can work from the single cell level, so from one cell to more cells, so 1,000, and this is reflected by also the fact that you can have also total RNA as input, not necessarily cells, and you can go as low as 10 picograms, which is the average quantity of total RNA of a single cell, up to 10 nanograms. So in that case, the SMART technology is employed and is using oligo-DT priming at the 3' end of the mRNA. So with the SMART-seq V4, you will produce double-strand cDNA libraries from messenger RNAs just because of the oligo-DT priming. So obviously this allows to avoid ribosomal RNA depletion because of oligo-DT priming, and you don't need to add further DNase treatment to your workflow because you will amplify only messenger RNA. All this happens in a single tube, and as I told you, it generates full-length cDNAs thanks to the SMART technology that for sure reaches the 5' end of the RNA. So this is here below an example of bioanalyzer trace for the double-strand cDNA libraries obtained with SMART-seq V4, and you can see first of all that you really produce full-length cDNA, so starting from 400 nucleotides to almost 11,000 base pair, so really full length. It's very reproducible and very sensitive because here maybe you cannot see it, but the blue line here is obtained with 10 picograms of RNA, so which means a single cell cDNA library. So it is very sensitive. So SmartSeq V4 produces a cDNA library that then will be prepared for sequencing. So of course, the DNA will have to be fragmented, and then adapters and indexes will have to be incorporated into the libraries. And so for this, we have several options, 2 for Illumina sequencing and 1 for Ion Torrent. So the protocol is fully compatible with Ion Torrent sequencing, as you can see here. And as far as Illumina sequencing is concerned, we have the option to go for Nextera XT protocol, which will fragment the DNA by tagmentation and also at the same time incorporate the adapter and indexes needed for Illumina sequencing. And a second option, if you have access to a mechanical fragmentation device, which can be a Covaris or a Bioruptor sonicator, you will sonicate your cDNA library and then use a DNA library preparation kit, such as our ThruPlex DNA-seq kits. I will come back later to those kits that basically will add the sequences needed for Illumina sequencing. How does it perform? Well, it performs really well. As I told you, this is the gold standard for the single cell analysis. We have more than 600 publications documenting the successful use of SmartSeq, and this is the last version, the version 4 of those kits. So you can see that starting from a single cell quantity of total RNA, we really dramatically increased the number of transcript identified, more than 14,000 with respect to the previous version. And this is due to all the tweaks optimizations we made to the protocol, such as incorporating locked nucleic acids in our template switching oligos, and this leads also to a reduced background, improved sensitivity, and reproducibility. This is shown here when we compared 2 replicates obtained at the single cell level, and you can see that replicate 1 against replicate 2 show really a very strong correlation indicated by the Pearson R coefficient, so 0.97. And most importantly, I would say, when you focus also on the lower part of this graph, so those are the transcripts which are slightly expressed, you can see that we further increased the correlations for these transcripts to 0.74 respect to the previous version of Smarter UltraLookit. So this to say that reducing the background, we also improved sensitivity and reproducibility really needed for single cell analysis or for ultra-low input amount of RNA. So in terms of performance, we can also have this kind of plot on the left, where we plot the gene coverage obtained by SmartSeq V4. So from left to right, we have the 5' to the 3' end of the transcripts, and you can see that you have really full gene body coverage obtained by SmartSeq V4, and obviously no bias in terms of coverage. So this is also very reproducible because all those lines here represent different single cell results, as well as increasing quantities of single cells, so 100 cells, 1,000 cells. So this is very, very reproducible. Just to mention that the SmartSeq V4 kit is also comes also in a very dedicated version, which is called designed for the Fluidigm C1 system. So you can increase a bit the output of the library preparation if you have this machine. And on the right side of this slide, I wanted also to mention that we specifically designed a SmartSeq V4 kit aiming at targeting the 3' end of the transcripts that we called SmartSeq V4 3' DE kit, just because sometimes customers don't necessarily need the full length information of the transcript and want to rather do more simple differential gene expressions. This is why it's called also DE, and thus focus on the 3' end of the transcript. So by doing 3' DE counting, you can have a really easier workflow, and most of all for bioinformatics analysis. So it works also at the single cell level, obviously, and we also specifically designed inline indexed oligo-DT, so adding indexes to these oligos to just increase the pooling capacity of the kit, and this is why you can pull up to 12 samples per Illumina index pair. So it leads to multiplexing of more than 1,000 samples. So those are two different versions of the kit, one which is really full length unbiased, and one which is more dedicated to 3' counting. We also recently launched a modified version of the SmartSeq V4 kit, and it's called the SmartSeq HT. So on this slide, you can see on the left the workflow of let's say the classical SmartSeq V4 kit, and on the right the SmartSeq HT kit. So HT stands for high throughput. So we dedicated this kit to higher throughput needs of customers when working with single cells. This kit is really validated using, for example, cells collected by fax, and as you can see, it is much more simplified. So you have only 3 steps for the HT kit compared to the SmartSeq V4. You have 6 steps, and it combines 1-step RT-PCR reaction. So basically, you save steps, you save time, and this is really needed if you want to increase your single cell throughput analysis. So the SmartSeq HT kit comes as a smaller format size of 96 run, just because of this higher throughput needs. So as I told you, SmartSeq ultra-low kits have been really documented in very high impact journals. As you can see here, have been tested with a lot of different mammalian models, circulating tumor cells, neurons, immune cells, stem cells, and so on and so forth, and also on a variety of non-mammalian RNA that can be plant RNA, it can be zebrafish, Drosophila, basically all the model organisms. So as you can see here, this is really thoroughly validated for almost any kind of cells and species. So moving on with the second part of our RNA-seq solutions. This leads me to degraded RNAs. So this is part also of what we call challenging samples, because as you know, sometimes you can face RNA of very poor quality, and we have solutions dedicated for that. This is what we call the smarter stranded RNA-seq kit. And so you can see on the right the workflow that we have for dealing with these degraded RNAs. So you already saw the global workflow of this. This is based on the smart reaction that I presented you before. Few optimizations and differences here have been made to deal with degraded samples that can come from FFPE samples, laser capture microdissected samples that can be prokaryotic RNAs. So to deal with that, instead of oligo-DT priming of the reverse transcription, we use random priming. So this allows to process RNA of almost any quality with a RIN as low as A3, so quite degraded. Obviously, it will work perfectly with high quality RNA. And the whole workflow of those stranded RNA-seq kits is very fast, because it's roughly 4 hours, and it incorporates in the workflow the integration of adapters and indexes for Illumina sequencing in a ligation-free manner. They are incorporated by PCR. These kits are called stranded, so it means that they will retain strand information on which strain of the DNA was produced the mRNA. And I reported here the quantities needed as input for those stranded kits. You see that it's very low. It starts from 10 to 100 nanograms of total RNA. So starting from total RNA, obviously you will have to deplete ribosomal RNA that can contaminate your sequencing data. And so for this, we have this product called Ribogon mammalian. It is meant for depletion of ribosomal RNA from mammalian origin, which means human, mouse, and rat. And this is how it performs in combination with our stranded kits. You can see here that the sequencing of our RNA is very, very low, below 1%, indicating an almost full depletion of ribosomal RNA. And in fact, it it translates to the good sequencing metrics that we observe with the stranded kits, with a very high number of genes identified, as well as a very good reproducibility here shown on the right across a wide range of input amounts, so as low as 100 picogram of ribosomal RNA depleted RNA up to 100 nanograms. So the correlation is almost perfect here. So when you will face even smaller amounts of RNA within your samples, this is what we call pico amounts of RNA. We also have dedicated solutions, and this is called the smartest stranded total RNA kit, pico input mammalian V2 kit. And so pico amount means that you can go as low as 250 picogram of total RNA. Very importantly, this kit is also really dedicated to strongly degraded RNA with RINs as low as 2. And this is particularly relevant for samples coming from FFP, for example, where you have a lot of degradation and you have a very small amount of RNA that you can retrieve from your sample. So how does it work? Well, it's almost the classical smart workflow that we already saw, with minor modifications, taking account of the very small amounts that we have at the beginning. So starting from total RNA, we will random prime the cDNA synthesis of all the total RNA, which means also of the ribosomal RNA. So after the completion of the reverse transcription, we will have a first step of amplification of the cDNA, because the input quantities at the beginning were very low. So we increase the quantity of the material that we have, and the rRNA depletion will not be done at the beginning of the workflow, but at this step here. So we will deplete the cDNA produced and coming from ribosomal RNA. And this is done with proprietary ZEP-R enzyme and R probes that will selectively degrade the cDNA coming from ribosomal RNA. So once the sample is cleaned of this, the library construction goes on, and the whole process is performed in roughly 5 hours. I spoke about really degraded samples, for example, coming from FFP samples. This is an example. So on the left, you have bioanalyzer traces of RNA that come from several FFP tissues that we tested, and basically you see that it's very degraded. We use those RNAs as input for the smartest stranded Pico V2 kit, and you can see on the left the sequencing library that has been produced, which is almost perfect. So showing this beautiful bell-shaped library here. So just to mention that this kit is really adapted to very low input amount and also very degraded samples. Those are sequencing metrics showing that starting from FFP RNA, you have very good correlation in if you compare different amounts of input RNA. So you have close to 1 of Pearson correlation for all the samples that we tested, and you also have minimal rRNA contamination, as we saw using ZAPR and R probes. This is visualized here in purple. Sorry. So the kit is really adapted to those degraded samples. Is very reproducible and sensitive. This kit also has been validated to work directly with the cells as input. It's been validated starting with as low as 50 cells up to 1,000 cells, and you can see that the sequencing metrics are very equivalent to what we just saw, so very high correlation and really minimal rRNA contamination. So smarter stranded PICO-V2 has been also widely published using samples as diverse as coming from FFP, has been published with cell-free RNA coming from plasma samples, cells isolated by FACS, isolated with laser capture microdissection, working directly on nuclei. So again, here the kit is strongly validated with a lot of different compromised samples. Another kind of challenging sample can be, for example, looking for biomarkers in plasma, and you may want to have a look at small RNAs and micro RNAs. So we have a solution dedicated for that, and it's called Smarter Small RNA seq for Illumina. You can see here that the workflow is based on the SMART technology, and it is dedicated to the study of small RNAs, which means from 15 to 150 base pair that include microRNAs, and it's the most sensitive kit on the market right now, because you can use as low as 1 nanogram of total RNA as input. Very important, this is a gel-free protocol, which means that you don't have to size select by electrophoresis your small RNAs, which can lead to bias in the library preparation. And of course, as usual, we incorporate adapters and indexes for Illumina sequences with a ligation-free approach. So you can see here how it works. Starting from total RNA, we will polyadenylate the RNA and induce the priming with oligo-DT primers. And then in the further steps, we will add by PCR the adapters and indexes for sequencing. So this kit has been used also for very challenging applications where RNAs may be very, very fragmented, below 100 nucleotides, for example. And this is the case, for example, like RNA immunoprecipitation coupled to sequencing, clip sequencing, as well as ribosomal profiling. How does it perform? It performs really well compared to methods using adapter ligation. Remind you that we use PCR incorporation of adapters and indexes. As you can see here, that we detect in a synthetic library of microRNAs. We detect 50% of the correct expression level of those microRNAs compared to 22% for adapter ligation method. So we really increased the correct identification of expression levels of microRNA. And we also have very high correlation. So for replicates and also across various input amounts of total RNA. It's been tested also, as I told you, as a proof of concept of biomarker discoveries on human plasma and serum samples. So using plasma and serum, we performed microRNA detection, and you can see high correlation for replicates in terms of microRNA detecting either in plasma or in serum for very low input amount of material, as well as a very high correlation, which is almost perfect. So it's really, in that case, suited for example, what we could call clinically relevant samples. Another very important product that we have is aimed at detecting rare transcripts. So you may want to focus on a small set of transcripts and may be very interested in transcripts that are expressed at a very low level, that are that have splice variants. And for this, we have this kit called Smarter Target RNA Capture. It works, it is based on hybridization of biotinylated probes that will hybridize to exons of the transcript that you want to target. Then you will pull down those complexes with streptavidin coated bits, and then the workflow after this will follow a classical SmartSeq v4 for the generation of the cDNA library and for sequencing. So this is a very streamlined workflow, which is very short. You can target up to 100 genes of interest, and it's really dedicated to rare transcripts or for studies aiming at identifying alternative splicing isoforms or gene fusions. So we also have products aimed at what we call immune profiling, and I will start with products dedicated to T lymphocytes and TCR, so T cell receptors. So as you may know, the body and the cells from the immune system have to face tremendous amounts of pathogens, which bear a huge amount of different antigens. And to cope with that, cells from the adaptive immune system, so for example, T cells, need to recognize those antigens via the T cell receptors and need to adapt to this diversity of antigens. So they need to show also diversity in the TCR to recognize the diversity in the antigen. And this is referred to a process that is called VDJ recombination. So this is schematized here very briefly. So you have here a beta chain of the TCR receptor, which is, so for the sake of the space, very similar to the alpha chain of the TCR receptor. Forgot to mention that TCR receptor is a heterodimer, seen here at the bottom left, composed of one TCR alpha chain and one TCR beta chain. And so the diversity observed in TCR is caused by these regions here, colored, called variable segments, diversity segments, and joining segments, and also with a constant region, which is constant in that case. So diversity is produced by somatic recombination. That means that all those segments will be rearranged at the DNA levels. Then upon transcription and splicing, these segments will further be rearranged to lead with millions of different combinations of the VDJ fragments for the TCR beta chain and VJ fragments for the TCR alpha chain. And the combination of alpha and beta will allow the T cell to detect a huge amount of different antigens. So how do we get inside to this with our solutions for sequencing? We have several kits for doing that, smarter human TCR profiling and smarter mouse TCR profiling. So on the right, you have the workflow, which is very similar, as you can see, to the classical smart workflow. And we use the smart technology really to get full-length information of the VDJ variable sequence I showed you in the slide before. And so this is performed like this. So you create the cDNA, and then with various primers, you will amplify TCR alpha and beta chain. And thanks to the smart technology, you will fully amplify the TCR region, which is variable. Those kits are really sensitive because you can go as low as 50 human purified T cells. It's a bit higher, 1,000 for mouse cells, and also as low as 10 nanogram of purified RNA if you want to work with purified RNA. The kits are very straightforward, as you can see from the workflow, very fast, and they generate also sequencing ready Illumina libraries. We also, just to finish on those kits, so these give you information on either alpha chains or beta chains, or the combinations of both chains in your samples, and it gives you also let's say the full repertoire of the clonotypes for TCR alpha and beta present within your sample, and this is what we call a population clonotype repertoire. So those kits are used for bulk studies. So to have more precise information on the TCR alpha and beta chain, we also have developed the smarter human single cell TCR alpha beta profiling kit. And contrary to the previous kit I showed you, it will give you at the single cell level the information of pairing of alpha and beta chain on a single cell, which is very important, for example, in studies where you want to track a specific combination of those chains in response to treatment during a pathology, but also to specifically monitor reactions against a very specific antigen, which is done by a very specific pairing of alpha and beta. So the workflow is basically the same as before. We just added few differences to render this kit usable at the single cell level, and this is mainly done with indexed smart oligo, as you can see here in pink, and you can pull in the end up to 96 different libraries coming from 96 different single cells. So how does it perform? It performs really well. I just put here a few comparison with other products from other vendors used for single cell TCR alpha beta pairing, so iRepertoire and 10x Genomics. And you can see here that the most important information is given here, so percent of cells that give useful data, so correct alpha beta pairing. We have the highest percentage of alpha beta pairing respect to the others, and again we get full length information of the TCR variable region. To finish with immune profiling solutions, I wanted also to mention that we have a dedicated kit for BCR profiling for mouse, so for B-cell receptor, that will allow you to, so following this classical workflow, to allow you to analyze separately or in combination the BCR-H, kappa, and heavy kappa and light chains. So this works also with directly with cells with low amount of B cells and low amount of total RNA, as we saw before for TCR profiling, and it can really be useful, for example, if you work with hybridomas, for example, to perform these BCR profiling studies. So this leads me to the second part of our portfolio for challenging samples. So the DNS-seq products that we have, two different technologies. The first one being the ThruPlex technology, dedicated for low and degraded DNA samples, and the Picoplex technology, dedicated for single cell genome amplification. So let's start with the ThruPlex. So ThruPlex is a technology for library preparation kit from low input and degraded DNA, as I told you. And as you can see summarized here is a very streamlined workflow. It just has 3 steps in a single tube, which is very important if you start from low input amount. You don't want to lose your samples. You don't have any purification steps in between. Again, you want to recover all your sample, and it's really fast because the whole process can be done in less than 2 hours with 15 minutes hands-on time. So this is really a very streamlined protocol leading to Illumina sequencing-ready libraries. So the ThruPlex chemistry works like this. So from DNA, you will have to ligate the adapters, which have this shape of a stem loop adapter. And you can see that ligation is made only at the 3' end, just to prevent the formation of adapter dimers. And then with just one PCR, we will resolve this nick here, open the stem loop adapters, and incorporate the adapter and indexes needed for sequencing. So this has been really optimized for dealing with low input and degraded samples, just by reducing the background at the level of the adapters and adapter chemistry, and also with the very easy and streamlined workflow. So ThruPlex kits come in 3 flavors. So for dedicated applications, if you want. So we have first the ThruPlex DNS-seq kits, which is what we call a multi-purpose library preparation kit. So it can be used, for example, for ChIP-seq and for preparing, remember, as I told you, libraries for RNA-seq downstream of SMART-seq v4. We have the ThruPlex Plasma-seq, which has been optimized for cell-free DNA coming from plasma. And we will see applications in a minute, for example, in non-invasive cancer research and also non-invasive prenatal testing. We have the ThruPlex Tag-seq, which on top of the ThruPlex technology incorporates unique molecular tags, and this is particularly useful for detecting rare alleles in cancer, for example. And you can see here quantities needed for input to these kits. So as I told you, one of the main winning application of the ThruPlex DNS-seq is ChIP-seq. We have a very high number of publications for this application. And so as far as cell-free DNA is concerned, so this is just an example of cell-free DNA research in cancer. As you know, circulating tumor cells can release DNA in the blood, and this is what we call cell-free DNA, or cell or circulating tumor DNA. And so to cope with this very specific type of DNAs, we have the Plasma-seq kit, cell-free DNA, and we also have the ThruPlex Tag-seq. So for very rare mutations and allele detection. So again, here a lot of references have documented the use of ThruPlex Plasma-seq for cell-free DNA analysis. And this is particularly relevant in cancer in the sense that all the ThruPlex kits are also validated with various target enrichment platforms. So it is perfectly suited for targeted sequencing. Listed here the platforms that have been validated with ThruPlex technologies. So for example, you see those classical Agilent SureSelect target enrichment systems. So if you want to do panel sequencing, exome sequencing, this is totally feasible downstream of ThruPlex. And just at the bottom here, you can see that ThruPlex DNS-seq kit in combination with target enrichment is really faster and more sensitive respect to the classical library prep kit of Agilent, for example, SureSelect. Sorry. So another application that can be done with through Plex Plasmaseq, I told you, is NIPT. So NIPT stands for non-invasive prenatal testing. So here you want to examine the fetal fraction of maternal plasma during pregnancy, and thus you focus on fetal DNA, and you want to look at, for example, mutations. You want to look at many other things like copy number variations and things like that. And again, through Plex Plasmaseq has been widely published for this kind of application. So the last technology that we have for DNA-seq is Picoplex, which is used at the single cell level. So this is the workflow of our Picoplex kits. So again, 3 steps in one tube, no transfer in between, and very fast. It accepts almost every kind of types of DNA, and of course single cells from 1 to 10, and it comes in 2 different solutions based on the downstream applications that you have. So Picoplex WGA. WGA stands for whole genome amplification. It produces libraries that then will be analyzed either by PCR or hybridization arrays. Or we have also a solution for those that want to use NGS for whole genome amplification analysis, and it's called Picoplex DNA-seq kit, which is compatible with Illumina sequencing. So on the right, you can see how Picoplex works. So it is based on whole genome amplification with quasi-random primers, and then so it produces libraries that can be used by PCR or arrays or for sequencing. As I told you, it works at the single cell level. It is really documented for a very high cell-to-cell and sample-to-sample reproducibility. It's one of the gold standards, I would say, for copy number variations analysis. If you want to see segments of DNA that are amplified or deleted, it's also very widely used for aneuploidy studies, so correct or not number of chromosomes. And so as you see here, and as I told you, you can choose your downstream application among PCR, arrays, and NGS. So here, just to summarize the fact that again, a high number of high-impact journals have published Picoplex for the main application that it has, so copy number variation and aneuploidy. So it has become the gold standard for pre-implantation genetic screening, so looking at CNVs and aneuploidies in PGS, but also it's been used for circulating tumor cells, where researchers looked at copy number variations at the single cell levels in these cells, and it's also used in this very innovative technology, which is called GNT-seq, so stands for genome and transcriptome sequencing, where from a single cell you both do transcriptome analysis as well as whole genome amplification, and picoplex has been used in this particular application. So proving the high performance and sensitivity of the picoplex kits. So with this, I'm done. You can, of course, have a look at our NGS resource portal, where you can find a lot of documentation, further documentation, tech notes, webinars, and posters. I indicated here also the email where you can write for getting information on our products. And with this, I'm done, and I'm gonna take questions if you have.